visible.
So, okay, thank you. So the topic assigned to me today is bioinformatics and its application in veterinary research. So as we all know, bioinformatics is still considered as an emerging discipline. A lot of evolutionary developments are taking place in the field of bioinformatics even today. And in a period of at least 1 hour, it is not possible to discuss all facets of this emerging science, bioinformatics, and particularly to highlight the application in the veterinary research. But I'll try to be very shortly describing the fundamentals of bioinformatics, the areas under bioinformatics presently blooming in different biological sciences, and also how we can apply the tools and techniques in our research efforts in the field of animal husbandry and veterinary research. So the term bioinformatics itself is not very old, and it was coined for the first time in 1970 by a scientist, Pauline Hogweg. But the context in which the term was used by him was not relevant to today's definition of bioinformatics. He referred to bioinformatics as a study of information processes in biotic system. This was published in 1970 in a journal called Chameleon. But later on, the modern definition of bioinformatics was given for the first time in 1987 by Dr. Hua Lin, and he described bioinformatics as a new subject of genetic data collection, analysis, and dissemination to the research community. But after the advent of the new genomic era and followed by the development of the omics technologies, there has been a lot of changes in the biological front, and there has also is associated with a lot of changes in the field of bioinformatics. And really speaking, the definition of bioinformatics is still evolving because of association and strengthening of different facets of the emerging science, bioinformatics, with the inputs from various branches of not only biological sciences, but also of various branches of material sciences. So today we consider bioinformatics is an interplay or an interface between material science and biological science. And a modern definition of bioinformatics says bioinformatics is an integration of mathematical, statistical, and computer methods between material sciences and the biological sciences. As has been rightly put up in a cartoon, which was published in 1990s in bioinformatics.org website, it was compared to the story in Panchatantra, wherein we know that 6 blind men were given the opportunity to touch an elephant, and thereafter they were asked, what is the morphology of an elephant? How did you find it? Then according to the part of the body of the elephant, the particular person touched, he described the structure of the elephant. The person who was holding the tail of the elephant, he said that the elephant is nothing but it's like a rope. Then the person who was touching the trunk of the elephant, he said that it is nothing like the banana tree. So everybody was trying to explain the structure of elephant according to his exposure. So bioinformatics has also become like that today because, as I said, there is involvement of various scientists of diverse background, starting from biological sciences to different branches of material sciences like physics, chemistry, computer science, engineering, statistics, mathematics, etc., to the field of bioinformatics. And therefore, everyone describes bioinformatics according to his own expertise or own exposure to the field. Say maybe for a physicist, the bioinformatics is concerned with the mostly the field of prediction of or determination of protein structure based on the X-ray crystallography. Similarly, those who are working with bioinformatics from the field of mathematics, for them the bioinformatics is nothing but the development of mathematical tools or certain mathematical formulae or algorithm to describe and interpret biological data. So we do not have a common definition of bioinformatics today. It is still emerging and also evolving with the involvement of various scientists with different backgrounds. Now in the omics era, we explain biological phenomena according to our concept of the different facets of omics technology. So in every strata of the omics technology, starting from genomics through transcriptomics, proteomics, metabolomics, and phenomics, one or the other way, the bioinformatics tools and techniques are involved or they are required to analyze and interpret biological data. Compared to meteorological world, the data that we obtain from biological experiments or from our survey and observation on biological system are very complex and diverse. And to analyze these diverse data, we need to have certain computational processes so that we can analyze them effectively within a short period of time and give our interpretation. That was the linkage established at the beginning with the advent of the genomics technology. After the gene sequencing technology was initially developed by Stenger, thereafter many sequencing technologies have come up, thereby it has increased our efficiency and the rapidity in sequencing. And now we are living in an era where we can get one eukaryotic genome sequence within very few hours. This was not true at the advent of the genomics technology, because when the Human Genome Project was started, it took around 13 years time to complete the sequencing of one single human genome. And it costed around $3 billion US. But the same can be done today by spending only around, say, $1,000 to $5,000. And also we can do it within a period of 1 to 3 days based on the platform of sequencing that you use. With the advent of the newer technologies, the protein sequences as well as the nucleotide sequences can be obtained from our biological experiment within a very short period of time. But the data that is obtained from these sequencing experiments are so enormous and huge that without the use of certain computational methods, it is not possible to analyze and interpret the data. So that was the beginning of bioinformatics. And that resulted in application and development of various tools and techniques to analyze biological data by using certain methods and principles of material science, like statistics, computer, then physics, chemistry, or even engineering. So that is the origin of bioinformatics. So therefore, the bioinformatics involves application of the principles, procedures, tools, and techniques of different material sciences to understand, analyze, and interpret biological data that we have obtained from our biological experiments. So there is application of bioinformatics now in all strata of our omics technology, starting from genomics through transcriptomics, proteomics, metabolomics, and phenomics. So without bioinformatics now, we cannot think of taking up any advanced research in biological sciences, including all applied sciences, also including the animal husbandry and veterinary sciences. So in the today's bioinformatics context, we have got certain most important areas wherein the bioinformatics is involved more prominently in biological sciences. They are the genomics, proteomics, drug designing, computational evolutionary biology, measuring biodiversity, and for modeling of biological systems. Now I think we have gone up to the extent of the synthetic biology as well as synthetic genomics, wherein bioinformatics can also play a vital role, not only modeling biological systems or biomolecules, but also it can now model organisms. So model organisms have already been produced, and synthetic genomics have advanced to a great extent after 2010. So the first and foremost advancement in bioinformatics started with the database designing and storage of biological information in the form of online and offline databases. Now we have a vast diversity of different biological databases according to the type of information or data stored, and they are mostly available online. And in fact, in today's definition of biological database, it has to be an online database shareable over internet, so that innumerable number of users can access the databases at a given point of time, and there can be free of cost sharing of data and information across the globe. Now we have got use of biological databases in all branches of biological sciences, including our own animal husbandry and veterinary sciences, particularly in the context of epidemiology and diagnosis of infectious diseases. Bioinformatics databases or online and offline resources available for the researchers can play a very vital role. So we shall discuss today some of those databases and how to access them and how they can be helpful for the epidemiologists and the veterinarians and medicals working in the field of infectious disease diagnosis and treatment. So there are many biological databases which contains a diversity of molecular information pertaining to genes and genome, proteins, as well as the literature. Of course, side by side, there are many literature databases available. And also, they also highlight not only the normal molecules or biomolecules and their structure, but there are also the defect in molecular apparatus in biological system that normally leads to certain diseases. As we know, the mutations may be related, or polymorphism may be related to disease susceptibility and also occurrence of the pathological abnormalities in animals as well as human beings. So not only the data pertaining to such mutations and polymorphisms in relation to disease resistance and disease occurrence are available in databases, but also many databases also highlight the methods and tools which can be used for disease diagnosis and susceptibility of host towards a particular disease, or for that matter, the susceptibility of the microorganisms to different kinds of antimicrobial compounds. We shall briefly discuss about some of them during the course of our discussion. So there are also databases which gives comprehensive description of the disease phenotypes. So phenotypic description of disease in relation to molecular variations in the genome can be also found out by accessing specific databases, and that can be used for diagnosis and treatment of infectious and non-infectious diseases of animals and human beings. Now, there are three important global databases which are considered as the most important resources for the biologist across different branches of biological sciences. Interestingly, all these three resources are created in three different continents of the world. One being at the central place, the GenBank, which is housed at the National Center for Biotechnology Information at in US, and the other one, European Nucleotide Archive. Formerly it was known as EMBL, European Molecular Biology Laboratory database. So this is housed in European Bioinformatics Institute, housed at England. The third one, most prominent, is DDBJ, DNA Data Bank of Japan, and it is housed in Japan, an Asian country. So they are hosted by three countries in 3 different continents of the world. But the beauty of these important databases is that they are interlinked to each other, and if one gets access to one, he can have access to the relevant documents and data pertaining to his interest in all different databases which are connected to it. So they are interconnected, and therefore mostly the scientists from European countries used to deposit their sequences in ENA or European Nucleotide Archive, and those from American continent, they usually prefer, or mostly even from the third world, mostly they prefer to deposit them in GenBank. But there is no problem in accessing the databases throughout the globe by entering into one of them. So from GenBank, you can also get access to the data from ENA as well as from DDBJ. As in October 2020, the latest statistics shows that the GenBank contains around 219.05 million sequences pertaining to different genes and genomes of a diverse type of thousands of different organisms. ENA contained around 2,829.1 million sequences, and DDBJ contained 2,566.18 million such information, sequence information in the databases. Online databases are also available, which can be used effectively for typing of infectious microorganisms. As all of us are related to disease diagnosis in one or the other way, either in the field condition as a field veterinarian or for confirmatory diagnosis in the microbiology, pathology, or other disciplines, we know that for final diagnosis of the causative agent, it is necessary not only to isolate and identify the organisms involved in it, but also to determine their specific biotype, serotype, or molecular type. So the molecular typing or biotyping has been now taking place by use of various databases which can help us in molecular typing of the microorganisms at a very rapid rate, and also it can be highly accurate. So almost 100% accuracy can be obtained for identification of a particular strain involved in a outbreak situation, and can correlate them to other strains or serotypes already known in the vicinity or elsewhere. So in surveillance of infectious diseases, and also outbreak investigations, and also in prevention and control measures, these databases can help a lot. And data pertaining to all our experiences with our work, both in surveillance, outbreak detection, and diagnosis of infectious diseases, can be stored and shared across the globe by uploading our information in the databases which are built for specific microbiological typing methods. Basically, today's rapid microbial typing methods include Plasmid profiling, Random Amplified Polymorphic DNA, or what we normally describe as REP-PCR technique, Field Gel Electrophoresis or PFGE, multiple locus sequence typing, what is usually described as MLST, similarly multilocus variable number of tandem repeats analysis, MLVA, repetitive sequence-based PCR, REP-PCR, ribotyping, and PCR ribotyping. Of course, this is not the exhaustive list. There are many other methods used, but most commonly used methods are Field Gel Electrophoresis, which is considered as the gold standard for identification of the molecular type of a particular pathogen in an outbreak situation or epidemic situation. Also, in certain cases, MLST or MLVA are preferred because PFGE is, although a robust technique, it is very accurate, but it takes around 5 to 6 days time to complete, and a very high level of skill manpower is required for doing the PFGE. But unlike that, MLST, or for that matter, the REP-PCR is comparatively a simpler technique which can be done very easily And that is why REP-PCR globally is gaining popularity nowadays, because it can be completed in around 3 hours, and it gives almost comparable results as that of PFGE. So for all these techniques, global databases have been built, and particularly there are some important global databases which give us access to the global information pertaining to the PFGE and MLST, and we can compare our results to the results already stored in databases to arrive at a conclusion, particularly regarding the pathotype or the biotype or serotype or the molecular type involved in a particular epidemic situation or an outbreak. So PulseNet is such a database which gives information pertaining to PFGE data. Similarly, ECMLST is a database which provides data pertaining to MLST of Escherichia coli. Similarly, for plant microorganisms also, several databases are there. I'm just quoting one such databases for plant microorganisms, which is called PAMDB, Plant Associated and Environmental Microbes Database. This is one platform emerging as a very popular and effective platform for the molecular biologist as well as epidemiologist of veterinary and human diseases, which is called CGE tools. So this is developed by Center for Genomic Epidemiology in Denmark, and there is a collaboration from global scientists through an effort from World Health Organization. So this platform has been developed as a very useful open access platform wherein you can provide an input the crude data that is obtained from any sequencing platform. Suppose you have got a sequencer, you are sequencing a complete genome of a particular microorganism, then without knowing anything about the sequence and assembly, analysis and interpretation, just the raw data that you obtain from the sequencer, the reads, what we call as reads, the raw reads can be uploaded to the site. Only thing is that you have to mention the organism, what is expected, or that means what is the presumptive identification based on the culture method, and then you have to provide the information pertaining to the sequencing platform that you have used. Suppose you have used Illumina platform, you should write Illumina. You have used some other platform, you have to mention the particular sequencing platform and then upload the raw data. Then within a few hours, through your email, they will give you the analytical report of the genomic sequence data given to them. So what are the analysis they will do? They will do everything. First, they will identify the organism correctly. They will identify the serotype. They will identify biotype or MLST type. They will identify how many different types of plasmids are there in the organism and how many these plasmids can be correlated to virulence or antibiotic resistance, and what are the antibiotic resistant genes present in the organism, what is their pathotype, whether they are pathogenic or not. All information pertaining to the particular organism will be given to you from one single analysis. So this type of whole genome sequencing analysis in future is expected to replace all other methodology that are in vogue today. So people are expecting that in next 3 to 5 years, maybe we will be not using the common molecular techniques for identification of microorganisms or their biotype or molecular type, but simply we will go for the whole genome sequencing, because whole genome sequencing is also becoming very easy and accessible and affordable. In European countries, you'll be surprised to know that now if you send microbial cultures in a lot, suppose you are sending 20, 30, that too with almost 100% accuracy. Now, another, so to say, tool that we can use in our research based on the whole genome sequences is provided by genome browsers. There are many genome browsers already established. They are single platforms providing not only the data pertaining to the genes and genomes of diverse eukaryotic as well as prokaryotic organisms which have been already completed, but also they provide you the common tools which can be used for analyzing those data and you can interpret. So these genome browsers have been developed both in US as well as in European countries, and there are many genome browsers which include genomic information of the livestock as well as in poultry. I'll give only one or two examples to them. So they not only store the information about the whole genome sequences of different livestock and poultry, including other eukaryotes and prokaryotes. Now they are in millions in numbers, and they also provide information pertaining to gene prediction and structure, the proteins of a particular organism, how many different variant proteins are there, what are the genes, location of the genes in the entire genome, and which gene is responsible for production of which protein, and the molecular variation among the genes, what are the polymorphisms or mutations already reported in the particular genotype. Similarly, they will also give you the expression profiles, transcriptomic results and analysis. They will also show the regulatory different species as well as between different strains or so to say breeds of the single species. These are some of the important genome browsers which can help you in analyzing the genomic sequences of livestock and poultry. Ensemble is one, UCSC genome browser, another. This is hosted in University of California, Santa Cruz. This is one of the most useful resources for the veterinarians also, because they also have got very good resources pertaining to the whole genome sequence, annotated information of different livestock and poultry, including the pet animals. Then genome maps, viral genome organizer. These are some examples of these genome organizers or genome browsers, which can help you in studying the genomic sequences and interpret your data by using tools which have been hosted by these genome browsers. This is the platform, the homepage of the UCSC genome browser. So you see here, I'm showing only a small part of the detailed phylogenetic analysis available, because they do have millions of whole genome sequences pertaining to thousands of different eukaryotes and prokaryotes. And based on that, by analysis, they have developed this phylogenetic analysis. Here you see, they have the whole genome sequence of pig, cow, bison, sheep, dolphin, then horse, dog, ferret, panda, and many others. So all can be found in one particular platform, and you can download information already uploaded by various scientists who have completed the genome projects of different animals. And you can, if you are also doing similar kind of analysis, you can have a comparative analysis of genomic resources within a short period of time. It cannot be only done in genomic level, but also if you are having small gene sequences, that can also be used for comparing the gene sequences of already known genome sequences that are uploaded to the online databases, genome browsers databases. Next is tools for data analysis. So for a biologist or for a novice who is not having much knowledge of the bioinformatics tools and techniques, it is not very tough today to learn bioinformatics and to play with the sequences that is generated in one's laboratory, because there are a lot many tools available online which are available with the detailed protocols or the procedure to be adopted for using them. so that you can learn by downloading the tutorials yourself. You can develop the capacity to analyze and interpret sequence data, maybe of protein sequence data, maybe of the gene sequence or genomic sequence data. So for that also, various kinds of tools and platforms are available online. So for assembly of the reads that we obtain from the sequencer machine, we can use assembly sequences, assembly sequencing tools. There are many such varieties. There are, for the same kind of analysis, you can have a number of different variable tools available. I'll go to some of the platforms which will provide such kind of tools. From them, you can easily either download or can use the tools online. XPaSy is one such common platform wherein thousands of different bioinformatics tools are available in the same interface. So when you are having sequence information pertaining to genes or genomes, or protein or proteomes, if you want to study any kind of, if you want to do any kind of bioinformatics analysis using your data, or you want to have comparative analysis of your data which already downloaded sequences from the databases, then you can easily use the online resources which are available in this platform. So this is developed by Swiss Bioinformatics Resource Portal, generated by Swiss Institute of Bioinformatics from Switzerland. So there are thousands of different tools which can be used successfully for doing any kind of bioinformatics analysis. The website is xpasy.org. Then another aspect of molecular analysis for understanding the interaction between the biomolecules, say between two proteins or between proteins and small molecules, what we normally refer to as ligands, we shall have to go for analyzing the interaction by way of molecular docking experiments. So molecular docking is another aspect in bioinformatics which can be done by using computational method which can predict the interaction between biomolecules, be it protein-protein interaction or be it protein-small molecule interaction. So such interactions can be studied based on certain physical principles. Here we apply the principles and techniques of physics and chemistry to analyze and interpret and predict the possible interaction between molecules or their affinity or their repulsion from one to other, so that we can easily interpret the interaction between molecules and correlate these interactions to the function of biomolecules. So for determining the metabolic function of any biomolecule, it is always necessary to look for the interaction the molecule can have with other biomolecules of the living system. So normally the successful interaction between molecules can take place only if there is a morphological similarity between the active sites of the molecule. So what we normally refer to as conformational similarity. The conformation has to be similar. It is similar to the lock and key relationship. A particular lock can be opened by a particular key only, because the ridges in the lock should match to the ridges on the key, then only it can be opened. It is almost similar. A particular protein can interact with another molecule of either protein or small molecule only if the physical conformity of the active surface of the protein molecule resembles in structure to the active compound.
Yes, sir, please.
So we were discussing about the molecular interactions. As I said, that the biomolecules interact when they do have some conformational similarity. If we consider a protein as the lock, then the other small protein or small molecule which will interact with that particular protein can be considered as a key. So there should be some conformational similarity in the active sites, then only they will interact. So this is a physical conformational similarity, and that is why the phenomena of protein-protein interaction or protein small molecule interaction can be described by using the principles of physics as is applied in material sciences. So by using the molecular mechanics, the application of physical principles can be done for understanding the interaction between biomolecules. So that is done in molecular docking to understand molecular interactions. But of course, our understanding of the molecular interaction cannot be completed if we simply consider that it is a physical interaction which does not involve any dynamism, which does not involve any change after the interaction has taken place. But biological molecules are very, very dynamic in nature, and even after interaction has taken place between two molecules, changes or some adaptation takes place thereafter also. It is not remaining static. So therefore, we can probably apply the, so to say, analogy hand in glove more properly than that of lock and key. As when we wear a glove, normally gloves are available in three sizes, small, medium, and large. Suppose I am using a glove pair of the medium size, but these are on standard sizes only, but that may not fit to my hand exactly. So what we do normally after putting on the glove, we used to move our fingers first so that we can adjust the glove to our fingers and the hand so that it appropriately fixes to our hand. So that is means after wearing also we adjust it so that it can fit, you can have the best fit between the hand and the glove. It is almost similar that the biomolecules, after they interact because of their conformational similarity, they undergo certain changes so that they can adjust their positions and can have the best fit between them, and therefore there will be molecular interaction taking place even after conformational, so to say, attachment between them. So that cannot be studied by simply using the principles of statics as we do in case of molecular docking. Therefore, another facet of molecular interaction study is involved, that is called molecular dynamic simulation. That means the dynamic relationship between two molecules can be studied by applying the techniques of dynamics that is used in physics so that we can understand the molecular interaction and the changes taking place between the molecules after the attachment taking place. Say, for example, in this particular number 4 figure. So here, the protein molecule, which is shown in the form of a helix, the protein molecule is binding to certain small molecules here. But after the binding has taken place by a static reaction, both the small protein undergoes certain changes, and therefore they will be undergoing conformational changes. Am I audible to you? Hello?
Yes, sir, you are audible, sir. Please continue.
Okay. So therefore, not only the molecular docking is necessary for understanding the intermolecular relationship, but also we have to proceed further by using molecular dynamic simulations, which is a very, so to say, computer intense program. So it needs high precision computer having high level of accuracy in calculations as well as very high quality speedy processors. So if you don't have such platform for studying molecular dynamic interactions of only 30 to 50 nanoseconds, it may take several days. So therefore, these computer intensive processes can be done only if you have got better computers accessible to you. So another aspect which is important for the biologists, including the veterinarians, veterinary scientists, is the designing primers for our molecular biology experiments for the PCR work. So specific primers can be designed by various online and offline tools, but today most of the important primer designing tools are available online free of cost. Mostly the primer tree is very commonly used, and primer tree has been embedded to Primer Blast software of NCBI. So if you go to the NCBI website, you get access to the Primer Blast. So which is one of the best platforms which can be used effectively for designing specific primers for your PCR reaction. So after designing the primers, you can also study the specificity and sensitivity of the primers by performing a predictive method which is called in-silico PCR. Means you can use the computational platform for studying the sensitivity and specificity of the primers design, so that before taking the primer pairs to your actual lab experiment, you can retest it for its sort of accuracy and sensitivity and specificity. So that can be done within a period of 2 to 3 hours, so that you do not fail in your wet lab experiment. So that reduces the requirement of time and cost for your molecular biology experiment. So that is why primer designing followed by in-silico PCR is now commonly done by using the online tools which are available free of cost. Open access softwares are available online. So you can also go for prediction of gene located within a particular genome sequence. What are the gene sequences, what are the coding sequences, what are the non-coding sequences, that you can predict pretty well. And also you can predict the gene function. You can identify the immunogenic components of an infectious agent, what are the important antigenic epitopes on the surface of the organism. You can also identify so thereby you can modify them, or you can apply specific methods so as to develop certain specific drugs which can interact with the receptor molecules so that it does not bind to the infectious organism. And these are the methods now used in molecular modeling experiments for modeling of proteins, identification of the active sites of the receptor molecules on the host cells, as well as for identification of the probable drug molecules among the candidate molecules. So I'll not go into detail about that, but these all functions can be done by computational methods using bioinformatics tools. Another aspect is the development of phylogenetic tree by use of phylogenetics and phylogenomics tools. So earlier, the phylogenetic analysis was mostly done based on certain conserved sequences. Particularly in case of bacteria, it was 16S ribosomal RNA sequences. In case of eukaryotes, mostly the mitochondrial gene sequences. But now, people have shifted from phylogenetics to phylogenomics. Means instead of using only certain specific gene sequences, people now take into account the whole genome sequence available for a particular organism in the databases. Because for most of the infectious organisms now, hundreds of variants of whole genome sequences are available in the databases, and that can be used very successfully for drawing phylogenetic interpretation and drawing the phylogenetic tree. So there are many online and offline tools available for doing this kind of analysis. For example, BEAST, MOSC, HiFi, Mega, POP. The Mega version 10 has become very popular among the students as well as the molecular biologists today, because this is freely accessible, downloadable from the website, and you can use it very effectively for analysis of data for drawing phylogenetic tree. And this is all the phylogenetic tree drawn based on the Mega 10 software is also acceptable for most of the high-rated journals of the world. So Mega is very popular nowadays among the molecular biologists for doing phylogenetic analysis. Then gene prediction, as I already said, that gene prediction is usually simple in case of bacteria, because in bacteria the coding sequences lie in the chromosome of bacteria contiguously. That means they are serially arranged without any intervention by non-coding sequences. So they can be easily identified by identification of the open reading frames. That means contiguously arranged codon sequences where they are introns intermittently, means between the coding sequences there are small stretches of the non-coding sequences interspersed between the coding sequences, and therefore we do not have a contiguous coding sequence. And the promoters are also located variantly from that of prokaryotes in case of eukaryotic genes, and therefore eukaryotic gene identification in a whole genome sequence is very, very clumsy and complicated. Only if you use appropriate tools, bioinformatics tools for that, you can identify the genes in a whole genome sequence. So there are many offline and online tools available for that. Say, for example, these are some examples only: Glymark, GenMark, and GenScan. Then another aspect is expression profiling. So transcriptomic analysis and proteomic analysis to understand the whole genome expression pattern or whole proteome expression pattern in one go can be done by use of sophisticated techniques available today. Say, for example, we can use the DNA microarray for analysis of transcriptome. That means the expression of the entire pool of genes in one go, in one method. Similarly, for proteomic analysis also, different methods are available, including the two-dimensional electrophoresis, 2D phase. So thereby, we can identify the proteins being synthesized in a particular cellular system at a given point of time. So under the influence of a particular condition. So these kind of whole genomic or whole proteomic analysis, giving a overall view of the expression profile, can be done only by use of certain specific tools. And these tools are also, again, dependent on certain specific bioinformatics tools and techniques for the analysis of the data. So there are high throughput EST sequencing platforms available for analysis of the occurrence of EST sequences in a genome sequence. Similarly, serial analysis of gene expression can be done by using a platform called SAGE, serial analysis of gene expression. Similarly, for analysis of the microarray data, there are different platforms available. They are called gene expression microarray analysis databases. And they can be used effectively to analyze and interpret the data obtained from microarray experiments, as well as from the 2D electrophoresis, as well as the spectrometric data analysis. We have got now different platforms of sequencing, as we already discussed, starting from Sanger sequencing. Now we have got the third generation sequencing technologies available in the form of the Pacific Biosciences sequencer or PFGE platform. Similarly, the Oxford Nanopore technology, ONT platform. Similarly, another SMART platform, which is commonly called SMART platform, single molecule real-time analysis. So these sequencing platforms have evolved in the recent past between 2010 to 2015, and they have made the sequencing very rapid and easy. And the error is also very, very less. And the length of the reads are also very high, along with the quantum of data generated per run. So out of the different techniques available today, popularly used, highly effective, high throughput sequencing platforms are the PFGE system and Oxford Nanopore Minion system. But of course, Illumina system, although it is little less, so to say, accurate and less efficient than that of PFGE or Oxford Nanopore, but even Illumina platforms are being used in most of the laboratories today, because this is also equally effective. And if you have got the Illumina platform available, the reads available from the sequencing data can be effectively used for whole genome sequencing analysis as well. Now, another type of bioinformatics tools are available for a biologist for antimicrobial resistance study. So there are specific databases and platforms available which provide not only data pertaining to the resistant genes and their function in different microorganisms, but also they provide the tools and techniques that are applicable for identification of the resistant genes and the resistome profile of pathogens. There is one such database called CARD, Comprehensive Antibiotic Resistance Database. So this is a curated collection of known resistant determinants. So this is very easy to handle, and you can identify the resistant genes in a whole genome sequence of microorganism by using this platform. So considering all these facts, you can easily now hope that the future prospect of application of bioinformatics in all different branches of biology, including animal husbandry and veterinary, is very bright. With the advent of the high throughput measurement techniques and the automation of the molecular techniques, I think it has become more rapid and more efficient today. And there is no denying the fact that now, without the help of bioinformatics tools and techniques, nobody can even dream of having advanced research in any branch of biological science. But of course, now, as I already referred to, the CGE tools or Center for Genomic Epidemiology tools are coming up, and in next few years, probably this kind of platform will give us one single platform for analyzing all information pertaining to infectious microorganism in one go. And that is also provided by World Health Organization free of cost. I think this will be very useful for the biologists, including the important barriers to use bioinformatics as of today is due to the lack of standardized formats, lack of common interfaces to the data, inconsistency in identifiers for biological entities, insufficient support for data exchange, as well as insufficient visibility. So there are many good tools not available free of cost, and at times it becomes difficult for workers having less resources to their access, find difficulty in getting access to very high costly platforms, and therefore most of the third world countries scientists are mostly dependent on the open access softwares and tools which are available free of cost online. So I think now the situation is such that most of the high throughput analysis can also be done by using open access softwares and tools. We need not worry about purchase of very costly, expensive softwares for bioinformatics analysis. So looking to the future, it is safe to say that bioinformatics is poised to change the world. Vancouver in 2013. I think it is very true in the current context of the use of bioinformatics in all facets of biological work. Now, so this is the platform that we have developed for molecular typing of Salmonella for determination of serotype. As I said, that more than 2,500 serotypes of Salmonella are available, and therefore in every advanced country there are certain national reference laboratories wherein they can perform the serotyping of Salmonella. It becomes very difficult for the molecular biologist or the microbiologist to get the serotype of Salmonella. But now with this platform developed, you can have a serotype of Salmonella done within 3 hours. So based on the 12 genes divergently placed in different serotypes of Salmonella, we developed this platform in collaboration with University of California, Irvine. So this was published in Indian Journal of Medical Research in 2017. So this is the platform that can be accessed through our college website. And anybody using this method can do simple PCR by using the primers designed by us and can test their results by using this platform online. Just they are to select which genes are positive, which genes are negative, and it will give you the serotype. And we have already included more than 99 important serovars of Salmonella which have been recorded in India to be prevalent in animals and human beings, and they can be identified within 3 hours instead of waiting for getting the results from the national centers, which usually takes around 3 to 6 months time. Another paper we have published in the high impact factor journal of biomolecular structure and dynamics. Current impact factor is 3.4. So this was based on our work on another important antigenic component of Salmonella, which was discovered in 2012 by a group of scientists from NICET, Kolkata. And thereafter, we found that the immunological capability of this particular gene, the product of the gene is known as the K2544, and the protein which creates is called PAGAN. And the PAGAN is antigenic. It was proved by both in vitro as well as in vivo experiments. But the real antigenic moiety of the PAGAN protein was not identified because the protein was not available in its structure, which was not crystallographed. And that is why in our laboratory, we predicted the structure of this PAGAN protein, and successfully we have designed the protein structure and also identified successfully 4 immunogenic epitopes on the surface of the proteins, and which were proved to be immunogenic. And thereafter, we took up this experiment in an in vivo trial by producing the recombinant protein in E. coli and thereafter testing it in mice. And that experiment has been done by one of my students, Dr. Girin Hazarika. He has completed his PhD on this line, and his paper has been already submitted for publication. Then another work we conducted on Clostridium difficile for molecular typing based on PFGE and ribotyping. So we found that ribotyping is more robust, rapid than that of the gold standard PFGE, and we found that certain ribotypes of Clostridium difficile is also present in pet animals as well as in pigs. And it was a very interesting story because, as we know, the Clostridium difficile is usually considered as a nosocomial pathogen, means it is a hospital-borne pathogen. Usually it is restricted to human being. It causes severe hospital-associated diarrhea and even cause deaths to hospitalized patients. Even in India, it has been already established that in many hospitals, the occurrence of Clostridium difficile may be to the extent of 10 to 15% in the hospitalized patients. And particularly it is caused by Clostridium difficile in patients who have been treated with antibiotics for treatment of the common diarrheal pathogens. So we found in our experiment that these pathogen, not only the pathogen, but also the specific biotypes which have been reported from human being can also be found in pet animals, in dogs. And some of the ribotypes which have been detected in human being have also been detected in pigs. So that have for the first time established the possibility of transmission of infection from pet animals as well as from pigs to human being. It may not be only acquired through hospitalization. And these papers were published, one in anaerobe, another is in molecular and cellular probes. This was the work done by one of my PhD scholars, Dr. Ismakul Hussain. So this is the result of the ribotyping. I will not go in detail. Then another work which was done by one of my master degree student, Dr. Purnima Gogoi, who is now doing PhD in IVRI. So she did a small piece of work, but interestingly, it was published in a highly coveted journal for microbiologists, the Journal of Clinical Microbiology. The current impact factor is 4.76. So it's internationally known as one of the most coveted journals in microbiology, published by American Microbiology Society. So this was based on the use of Field Gel Electrophoresis as well as a very rapid technique that we developed in our laboratory based on the repetitive element-based, repetitive sequence-based PCR, what is called REP-PCR. And by comparing these two methods for molecular typing of 12 known Salmonella pathogens, we could establish that the REP-PCR, which can be completed in 2 to 3 hours, can be almost comparable to the PFGE, which requires 5 to 6 days for interpretation. So this is a very handy technique now, which can be used in epidemic situation for identification of the molecular type of a pathogen. Another recent article that we have published after the COVID-19 pandemic globally, that was also published in the highly coveted journal, Journal of Biomolecular Structure and Dynamics, which involved identification of a druggable candidate in the form of a repurposed drug, which is already known as an antibiotic. We identified it by molecular docking and molecular dynamic simulation experiment. biomycin, which is already known as an antibiotic long before as an anti-tuberculous drug, can also be active against the main protein, protease protein of SARS-CoV-2 virus, which is the causative agent of the COVID-19. So this was published in Journal of Biomolecular Structure and Dynamics. Similarly, another work on SARS-CoV-2 virus, that is COVID-19, was also recently published in Virus Research, another high effector journal, that was on analysis of codon uses of severe acute respiratory syndrome coronavirus 2 and its adaptability in dog. So in this work, we have established that the humans are the favorable host for the SARS-CoV-2 virus, although it can also cause disease in canines. Particularly, it is reported from several countries already in dogs, in let me say your cat, as well as in lion. Already, it has been in tiger also. It has been reported. So in case of dogs, we found that the codon usage pattern of the SARS-CoV-2 virus is almost similar to that of the human being. And that is the reason that out of all the different pet animals or other carnivores, dogs are a preferred host for SARS-CoV-2 virus. Similarly, we also established beyond doubt that rather than the mutational process or the mutational pressure, the normal selection process is more important for the variability or diversity in the SARS-CoV-2 virus. And therefore, the overall mutation pattern that is already reported in the SARS-CoV-2 virus in different populations might not be having direct impact on the change of the pathogenesis of the pathogen so far. So this was also published in Virus Research. We predicted its structure and we also predicted its molecular function. Then another publication we had in Infection, Genetics and Evolution, based on our research in a group in collaboration with IBSD in fall and Institute of Life Sciences, Bhubaneswar. This was on molecular epidemiology of Japanese encephalitis virus in pig population of Orissa, Assam and Manipur states of India. This particular project was taken, and this has been already completed by one of my postdoc scholars, Dr. Iftikhar Hussain. It is already submitted to journal and it is in the process of acceptance. The last revision has been already made yesterday and we are hopeful of publishing it very soon. So here is the prediction of the molecular structure of the protein as well as the molecular dynamic simulation study of the haptameric complex of Clostridium perfringens epsilon toxin. Enterotoxin as well as epsilon toxin work has been already completed and in the process of publication. So these are the works done in our laboratory and bioinformatics in the recent past. Thank you very much